Welcome to the deep dive. You might think you know goldfish, those little orange guys swimming in bowls, but we're about to take you way beyond that.
Oh, definitely. There's so much more to them.
We've got some incredibly detailed information here looking at goldfish varieties and their genetics. And our mission today really is to uncover the truly surprising stories behind their evolution, how we domesticated them.
And just the sheer explosion of different forms they've taken. It's quite something.
Exactly. Get ready to have your perception of the humble goldfish completely transformed.
Absolutely. What's truly remarkable is that this fish, often seen as so, well, basic, belongs to a family, the Cyprinidae, that's the carp family.
Ah, carp.
And it's one of the great success stories of the fish world.
Okay, carp like those bigger fish you sometimes see in rivers and lakes.
Exactly those. And this family has some very specific traits, like they don't have teeth in their jaws, which is interesting.
No teeth. How do they chew?
Well, they have this special upper jaw that can pop outwards, sort of protrude, and the chewing happens further back with teeth located in their throat pharyngeal teeth.
Weird.
But the real headline here is just how many different kinds of fish are in this family. We're talking around 1,700 recognized species.
1,700. That's mind-boggling.
It makes the carp family the most diverse group of freshwater fish on Earth. By far.
Wow. Where do you even find that many different types? Are they everywhere?
Pretty much, yeah. Their natural homes cover a huge area, most of Eurasia, Japan, the islands of the East Indies, North America, and Africa.
So vast areas.
And interestingly, while they mostly live in freshwater, some can actually handle brackish, even salty water. Talk about adaptable.
So they're survivors, clearly. You mentioned they have ancient origins. How far back are we talking? Like dinosaurs?
Not quite dinosaurs, but still very old. The fossils tell us the carp family has been around for over 40 million years.
40 million.
And here's a really fascinating connection. You don't find them naturally in South America or Australia.
Oh, why not?
Well, scientists think this absence is linked to continental drift. The theory goes that this fish family evolved after the supercontinent Pangaea started breaking up.
Ah.
But the southern land masses, South America and Australia, had already drifted too far away by the time the early carp started spreading. It's all about timing and geography.
That's incredible. So the humble goldfish's ancestors are tied into the very history of our planet's land masses. Where did these ancient carps first show up then?
The strongest evidence points towards East Asia as the origin point.
Okay.
From there, different kinds of carp, like Cyprinus and Carassius. And Carassius is the group goldfish belong to. They naturally spread across Eurasia. But the global distribution we see today, that's largely thanks to us humans.
Ah, right. So we move them around the globe.
Precisely. And while this means goldfish are enjoyed worldwide now, it's been a bit of a mixed blessing.
How so?
Well, for instance, there's a good chance that even common carp weren't originally native to the far western parts of Eurasia, like Britain. Humans probably played a role there too.
Really? Even common carp?
It seems likely, but it's much clearer in places like North America and Australia, where European settlers deliberately introduced them.
And that's where the mixed blessing part comes in.
Yeah.
Get these unintended consequences, right? Like, wasn't there that famous rabbit plague in Australia?
Exactly that kind of thing. Carps and goldfish are incredibly tough and adaptable, so when they get introduced into new environments where they don't naturally belong, they can thrive, sometimes too well.
Right.
And in many cases, these feral populations, fish that have escaped or been released and are now living wild, can negatively impact the local wildlife and upset the natural balance. It's a real problem.
So you end up with strict rules about introducing non-native species.
You do,
Ah.
because these aren't just a few escaped pets. They can really establish themselves and cause significant ecological issues.
You mentioned some interesting features cropping up in these wild or feral goldfish populations. What's the story there?
Yeah, one particularly intriguing example is the bluebelly goldfish found near Lake Erie in Ohio.
Bluebell.
Yeah, this type has less of that shiny stuff guanine on its scales, which gives it a different look. And interestingly, similar-looking goldfish have been seen elsewhere. They've been described as net-like transparent in the Far East and mock metallic in Great Britain.
Huh. So similar mutations popping up independently.
Possibly, or related ones anyway. And the fact that this bluebelly is relatively common in the Lake Erie area is likely due to what's called the founder effect.
Founder effect. What's that?
Imagine just a small group of goldfish, maybe just a few, being introduced to the lake, and some of them happen to carry this slightly unusual trait.
Okay.
Because they're among the first to reproduce there. That trait just by chance, can become much more common in all their descendants in that specific location compared to the original population they came from.
That's wild. So these accidental introductions are like little natural experiments in evolution happening right before our eyes. Now, were scientists always this interested in goldfish, or is it more recent?
Well, they weren't always the main focus, maybe, but they certainly caught the eye of some important thinkers pretty early on. Back in the 19th century, Charles Darwin himself mentioned goldfish in his book Variation of Animals and Plants under Domestication.
Darwin knew about fancy goldfish.
He did. And a bit later, over a century ago now, William Bateson, a key figure in early genetics, saw them as a really promising subject for studying how different characteristics arise and are passed down. He noted some things that are still relevant today.
So even back then, they were recognized as a good model for understanding heredity.
Precisely. But the really deep, in-depth study of goldfish genetics took off in the 20th century, particularly in the Far
Right,
East.
where they originated.
Exactly. Scientists like Shisan Chen in China and Yoshiki Matsui in Japan did some groundbreaking work. They essentially established that the fancy traits we see, the different shapes, fin types, colors, follow Mendelian rules of inheritance.
Mendelian, like Gregor Mendel and his peas. Those predictable patterns.
Yes, the fundamental principles are the same: dominant genes, recessive genes, how they combine. However, understanding the full picture in goldfish wasn't straightforward. It wasn't really until after World War II that a crucial discovery was made about their basic genetic makeup.
Oh, what was so unusual about it? Did they find some weird extra genes or something?
Even more fundamental than that, it turns out goldfish, along with their close relatives, the common and crucian carps, have double the number of chromosomes compared to most other members of the carp family.
Double. How many are we talking?
Most carp have around 50 chromosomes, right? Goldfish and these close relatives have 100.
Whoa, 100 chromosomes.
Right.
Double the genetic material. What does that actually mean for a goldfish?
It's a condition called polyploidy. In this case, it's likely tetraploidy, meaning they essentially have 4 sets of chromosomes instead of the usual 2 sets that most animals, including us, have.
4 sets, like having backup copies of everything.
Sort of, yeah. Think of it like having backup copies of many of their genes. And this duplication has had a huge impact on their evolution.
How so? Good or bad?
Well, both in a way. On one hand, it provides a richer genetic toolkit. Having extra gene copies means some can potentially take on new functions over time without losing the original function. This can fuel increased diversity and the potential for more rapid evolution.
So it's like having extra ingredients in the evolutionary recipe, allowing for more experimentation.
That's a great way to put it, exactly. However, having all those extra chromosomes also makes understanding their genetics much more complicated for us scientists.
Right, I can imagine.
Simple Mendelian ratios, like that classic 3-to-1 pattern you see with single genes in diploid organisms, well, they get altered in polyploids.
So the math changes.
It does. For instance, a trait controlled by a single gene might show up in something like a 15-to-1 ratio instead of 3-to-1. This added complexity has made it more challenging to fully unravel the genetic basis of all the incredible variety we see in goldfish. It's not impossible, just trickier.
Okay, so the extra chromosomes are a powerful engine for creating diversity, but they also make the genetic rulebook a bit harder to read. Now, how does all this genetic potential actually translate into the physical differences we see? Like, how do you get a fish with twin tails or telescope eyes?
Right, that's where we connect the genetics to how the fish actually develops, its embryology and growth. Some traits appear very early. The twin tail phenotype, for example, which you see in fantails and rukins and so on, that becomes visible while the fish is still an embryo. But many other fancy features, those amazing telescope eyes, or the little pom-pom-like nose growths, or the fleshy head growth on lion heads and orandas they develop after the fish have hatched, they're post-embryonic developments.
So different fancy bits appear at different stages of life. It's not all there from the start.
Precisely. Scale transparency, for instance, that shows up pretty early, but those bulging telescope eyes, or the upward-gazing celestial eyes, they develop later, sometimes over the first year. And the characteristic head growth, the hood, that's often one of the last major fancy traits to appear and develop fully.
Interesting. What about color change? From that wild brownish color?
Yeah, the change from the wild bronze or olive color to the reds, oranges, and yellows we admire also happens at different times for different fish and different colors. It's quite variable.
But some fish start black. Right, like the Moor.
Exactly. Some color variations, like the intense black of a Moor or the chocolate brown of some specific breeds, don't really involve a color change from that wild type in the same way. The blacks just get darker and more intense, and the browns seem to have their unique coloration right from the start or very early on.
So they're kind of born with their signature look in those cases. Okay, you mentioned something really wild earlier, gynogenesis
Mm.
in Carassius. That sounded like science fiction.
It is quite remarkable, isn't it? Dinogenesis is a type of reproduction, and it seems to be linked to those higher levels of polyploidy we talked about fish, with even more than 4 sets of chromosomes, maybe 6 or 8 sets.
Hexaploids, octoploids.
Okay. In some species of Carassius with these higher ploidy levels, it seems to be their normal way of reproducing. What happens is the egg cells don't undergo the usual process that halves the number of chromosomes. So the egg ends up with the full set of chromosomes from the mother,
All of them.
all of them. Then these eggs still need a little nudge, let's say, to start developing. And that nudge comes from sperm, usually from a related species. But here's the key part. The sperm only activates the egg, doesn't actually fertilize it in the normal sense.
So no genetic mixing.
Correct. There's no joining of the sperm and egg's genetic material. As a result, all the genetic information in the offspring comes entirely from the mother. They're essentially all female clones of the mother.
That's incredible. A fish species that can essentially clone itself, just using sperm as a trigger. Nature really is full of surprises.
Wow. Okay, before we get lost in all these amazing variations, can we rewind a bit? What did the basic goldfish morphology look like? The original undomesticated fish.
Right, good point. The ancestral goldfish, the wild Carassius auratus, had a fairly streamlined body, kind of flattened from side to side.
Okay.
Smooth curves along its back and belly. Its width was maybe about 2/3 of its depth. The head and the deepest part of its body were each roughly 1/3 of its total length. Pretty balanced proportions.
Any other key features?
Yeah, the line running along its side, the lateral line, was almost straight. And its fins had specific proportions too.
Mm.
The tail fin, the caudal fin, had its top and bottom lobes pretty much equal in size.
Uh-huh.
The notch, the indent between the lobes, was about half the length of the lobes themselves, and the lobes were maybe about 1/4 of the body length, so nothing too dramatic.
So a fairly standard, efficient fish shape. Exactly. It's really important to keep this original form in mind because the incredible diversity we see in goldfish today just shows how much domestication, how much selective breeding by humans has altered these basic features.
It really does. Going from that sleek, simple body to fish with huge humps, flowing fins longer than their bodies, bulging eyes. It's quite a transformation.
Yeah.
Now let's get into that domestication process itself. How did we even get from those wild carp-like ancestors to the goldfish we have in tanks and ponds today?
Well, our current understanding, pieced together from historical records and scientific studies, suggests a kind of 3-stage process.
Okay, 3 stages.
First, there's an initial period, maybe call it pre-domestication, where they weren't truly domesticated yet. Then the actual domestication process kicks in. And finally, the diversification stage, where all the different varieties start to emerge.
And figuring out the exact when, where, who, how, and why. That's the tricky part.
It is a bit of a puzzle, yeah.
Yeah.
But we have some pretty strong clues.
So that pre-domestication phase, what was happening then? Were people just noticing some unusually colored fish swimming around?
That seems likely, maybe seeing occasional gold or yellow variants of the wild carp, but the real turning point, the actual start of domestication, appears to have happened during the Song dynasty in China.
The Song dynasty.
Yeah.
When was that, roughly?
Around the 10th to 13th centuries AD. After a period of division, the Song emperors reunified the empire. And during this era, there was a really significant change. Goldfish started being brought into private ponds in people's homes and gardens.
Ah, okay. So moving from, say, public waters or maybe temple ponds into people's personal spaces.
Exactly. Before the Song dynasty, during the earlier Tang dynasty, the rise of something called ponds of mercy near Buddhist temples seems to have played a key role.
Ponds of mercy.
Yeah, these ponds house various aquatic creatures that people would release there as an act of compassion, and they would have included naturally occurring color variations of carp.
So people were already kind of collecting these unusual fish in one place.
Right. Having these golden or silvery fish gathered in such culturally significant places likely led to people starting to value them, maybe protecting them. It's like an accidental early step towards selection.
Like an unintentional cultivation process almost.
Precisely. But the Song dynasty really marks the shift to deliberate domestication, with goldfish being intentionally kept and probably bred in private ponds. One key researcher, Chen, believed that the establishment of these private ponds likely happened during the Southern Song period, specifically in the city of Hangzhou.
Lion cow.
Because ponds of mercy had existed there for a long time, so there was likely already a culture of appreciating these fish. And we even have records of imperial interest, Emperor Chao Ku, for instance.
Mm-hmm.
He seems to have been a real goldfish enthusiast. Just before he died in 1186, he actually ordered the collection of gold and silver fish specifically to fill his own ponds.
Wow, the emperor himself.
Yeah. And royal interest like that could very well have strongly boosted the popularity and development of goldfish keeping among the wider population. It gives it a certain prestige.
Definitely. So royal enthusiasm really helped fuel that early goldfish craze.
Mm.
Okay, so from those early ponds in Song Dynasty China, we've now got this incredible variety of breeds. It's almost hard to keep track.
Mm-hmm.
Are new types still being developed?
Oh, absolutely. The development and evolution of new goldfish varieties is very much an ongoing process. These days, it's happening primarily in China, actually.
Still in China after all this time?
Yeah, they're really at the forefront now. Many of these new forms are, variations on existing themes, combining traits in new ways. But hopefully, with a better understanding of genetics, we can not only protect the diversity we already have, but also maybe expand it in more informed ways in the future.
It's just amazing to think that this whole process, this careful selection and breeding based purely on what humans find attractive, has created such a huge range of forms from that single, relatively plain ancestor.
It really is a testament to the power of artificial selection.
Let's talk about some specific varieties. The Shubunkin, that's a relatively recent one, isn't it?
Yeah.
Compared to some others.
Yes, the Shubunkin is a 20th century creation. It originated around 1900 in Japan. Research suggests it came from breeding the walk-in.
The walk-in, what's that like?
The walk-in is a more basic, long-bodied, twin-tailed goldfish. So they crossed the walk-in with a Chinese calico demican.
Demican, is that the telescope eye?
Yes, the demican is a telescope-eyed goldfish, often calico-colored. Both of those parent types already had multiple colors. The walk-in showed red, black, bluish, white. The Demican had black, red, purplish markings.
So you mix those two.
Right. When they were crossed, the offspring were a mix, as you'd expect. Some looked like wild goldfish, some like the Demican parent, some like the walk-in. But about 20% showed the desired traits, the single tail of the walk-in ancestor, but the transparent scales and mottled colors of the calico Demican.
Ah, so they selected those ones.
Exactly. Those were then carefully bred together to establish the Shubunkin type and create new color patterns within it. Interestingly, its Japanese name translates roughly to vermilion red dappled with different hues, which is quite descriptive.
And we use the Japanese name in English.
We do, which is a bit unusual, but it's stuck. It's become very popular in the West.
So it's known for that mix of colors and patterns all on one fish. Okay, what about the comet? That seems like a classic American goldfish.
The comet's origins are indeed traced back to the U.S., specifically to fish ponds managed by the U.S. Fish Commission in Washington, D.C. in the 1880s. It was first sold commercially around 1883.
And what makes it a comet?
Well, its body shape is similar to the common goldfish, but the comet is noticeably more slender, more streamlined. And its most defining feature is the significant elongation of all its fins, especially the tail fin and the anal fin underneath.
How long are we talking?
They can be as long as or even longer than the fish's body. Very dramatic, flowing fins. It's considered a great fish for ponds because it's hardy, it swims very gracefully with those long fins, and it tends to breed pretty true to type.
Those long, flowing fins definitely give it a very distinctive, elegant look. Okay, now, the more the black, velvety goldfish with the big, protruding eyes, where did that one come from?
The Chinese Moor telescope is a really striking fish. Early descriptions suggest that in older individuals, the fins could become quite long and flowing, almost like a veil tail. We see that today in the modern broadtail Moor variety.
Right.
A good Moor provides a wonderful visual contrast to the brighter metallic and calico varieties in an aquarium or pond. The black color can be incredibly deep, so much so that it looks velvety and completely hides the natural shine of the scales underneath.
Is it always pure black?
Usually it's black all over, except maybe on the lower belly, which might sometimes show a bit of blue-gray or even a golden-brown tinge. But the ideal is that deep, uniform black.
And breeding them
Yeah.
is straightforward. Black plus black equals black.
Interestingly, no. Breeders have found that the best way to get a high proportion of black offspring isn't necessarily by breeding black moors with other black moors.
Really?
Yeah, they often get better results, a higher percentage of good black offspring, by pairing black telescope goldfish with red telescope goldfish.
That's surprising. So sometimes mixing seemingly opposite colors can actually concentrate the desired one.
Huh.
Okay, what about the celestial telescope? Those are the ones with the eyes that look permanently upwards, right? They always look a bit startled.
Yes, the celestial. One well-known expert, William Innes, considered the celestial to be one of the trickiest goldfish varieties to keep and breed successfully. They are quite delicate.
And the eyes.
Yeah.
How did they get like that?
It's a developmental process that happens in two stages. First, the eyes start to bulge outwards, just like in other telescope varieties, like the Moor.
Tuh.
But then the pupils gradually rotate upwards, eventually pointing almost directly towards the top of the head. This unusual eye development isn't caused by anything external, like disease. It's a specific trait they inherit.
So it's genetic.
Absolutely. Early drawings show a fish with a short, somewhat egg-shaped body and a tail about the same length as the body. But the main focus, the defining characteristic, was clearly those unique upturned eyes.
Yeah, they're certainly unforgettable. Now let's talk about the lion head. That's another one with a very distinctive head feature.
The lion head, yeah. The name comes directly from the Japanese shishigashira, which literally means lion head. It's known primarily for two things: that fleshy, raspberry-like growth over its head and gill covers.
The hood.
The hood, yes, and the complete absence of a dorsal fin. Plus, it typically has a short, thick body and relatively short fins compared to some other fancy varieties.
Does that head growth cause any problems for the fish?
It was once thought that the hood growth might slightly interfere with their breathing, suggesting they needed really well-oxygenated water, especially when it's warm. But well-bred fish seem to manage fine.
And the fins.
Like all twin-tailed fancy goldfish, the lionhead should ideally have double tail fins and double anal fins. Though some early descriptions were a bit lenient on the finnage if the head and body shape were exceptionally good.
What colors do they come in?
The common colors historically were red and white, or combinations, but pure white bodies with pale yellow heads were also noted, and lion heads with transparent scales, like shubunkins, have also been bred, though they're not very common.
And they definitely don't have a dorsal fin. I remember reading somewhere that people used to think you could make them dorsal-less.
Ah, yes, there was an old incorrect idea that you could create a dorsalless fish simply by removing the fin in young fish, but that was dismissed pretty early on. Acquired characteristics like that aren't inherited. You can't pass on a surgical modification to offspring. The lack of a dorsal fin is purely genetic.
Right. Makes sense.
All right.
You also mentioned the name ranchu.
Yes, in Japan, the name ranchu has become widely used, often interchangeably with lionhead, especially for the fish developed and bred to Japanese standards. There are subtle differences often debated by enthusiasts, particularly in the curve of the back.
So the name itself has a bit of a complex history and regional variations. And that fleshy head growth is the absolute key feature.
Okay, what about the fringe tail or ryukin? Those seem to have a lot of variation, especially in their tails.
The fringe tail, or ryukin as it's often called, especially the Japanese type, is really important historically because the veil tail was developed directly from it by breeders in America, particularly in Philadelphia.
Ah, so the veil tail came from the ryukin.
Yes. The tail fin is actually used to distinguish two main types within the broader fringe-tail ryukin category. You have ribbon tails or swallow tails, which have deeply forked tail fins.
Okay.
And then you have broad tails or veil tails, which have a much less pronounced split or indentation in the tail fin. It's more squared off, like a veil.
And the ryukin specifically, what's its main characteristic now besides the tail? While it wasn't always emphasized in orbiter writings, the main difference between modern ryukins and modern veil tails, besides the tail shape subtleties, is their body profile. The ryukin tends to develop a noticeable hump on its back, right behind the head.
Right, that shoulder hump.
Exactly. Whereas the ideal veil tail should have a smooth, continuous curve along its back with no hump. Although some veil tails might still show a slight tendency towards developing a hump, which kind of hints at their ryukin ancestry.
So the veil tail is essentially a sleeker, smoother-backed version of the ryukin, but keeping or even exaggerating those incredibly long flowing fins. It's amazing how breeders have focused on and refined different features over time.
It truly is.
You mentioned those key figures in goldfish history earlier, Chen and Matsui. Their work must have been incredibly important for understanding all this variety.
Absolutely fundamental. The information we have on goldfish really comes from two main types of sources. You've got the general guides on how to keep them, which often don't go into huge detail about all the specific varieties.
Right, the basic pet store guides.
Yeah. And then you have more in-depth works, like those by Wolfe and Smith in the early 20th century, which were very valuable at the time for describing the different types known in the West. Between the two world wars, a book called Goldfish Varieties by Innes was hugely influential.
Okay.
But the real quantum leap forward in our detailed understanding came from the work of those two scientists from the Far East. Shisansi Chen in China, publishing in 1925, and Yoshichi Matsui, a Japanese geneticist, publishing a series of papers in 1934.
Chen and Matsui.
Their contributions are absolute milestones. Chen's 1925 work, Variation in External Characters of Goldfish Carassius serratus, remains, even today, arguably the most detailed and comprehensive description of the sheer range of physical variations across all goldfish varieties. It's incredibly thorough.
And this was back in 1925.
Yes. And while people in the West suspected they hadn't seen the full range of Chinese goldfish diversity, it actually took another 30 years or so after Chen's publication for many of the types he described to actually appear and become known in the West.
Wow. So Chen's work was like this hidden treasure map of goldfish diversity that only gradually got explored by the rest of the world.
That's a good way to think about it. Interestingly, Chen didn't give formal descriptions for each named variety like a modern standard. Instead, he discussed them while focusing on specific variable features like fin types or eye types, which makes comparing his observations with other writers quite interesting.
So he was looking at the building blocks of the varieties. What are some of these specific physical traits that Chen and Matsui studied in such detail?
Well, one interesting example Chen described is the outfolded operculum.
Operculum, that's the gill cover.
Exactly. In this variation, the hard gill covers curve outwards, away from the body, exposing the delicate red gill filaments underneath.
Is that desirable?
Apparently, Chinese breeders found this trait interesting, perhaps novel, but in the West, it's generally seen as a defect or deformity, and is strongly discouraged in breeding and showing.
Okay, different tastes. What else?
Chen also did a very careful study of the colors of metallic-scaled goldfish. He even illustrated the different types of pigment cells he observed in their scales.
The microscopic level.
Yes. He showed, for example, that the xanthic forms, the reds, oranges, yellows, basically lack the dark melanin pigment. Blue forms lack the orange-red pigments. Black forms, like the Moor, have so many dark pigment cells, melanophores, that they completely hide any other colors underneath.
And brown.
Brown fish, he found, have both dark melanophores and the orange-red xanthophores present. And in all these metallic-scaled fish, the shiny, reflective appearance comes from special cells called iridophores, which contain guanine crystals.
Guanine. That's the shiny stuff.
That's the shiny stuff. He also noted that the paler scales on the belly simply have far fewer pigment cells overall, even though the individual cells might be a bit larger, and the underlying bluish tissue can be more visible there.
So different combinations of these few cell types create the whole palette.
Exactly. Silverfish result when both the red yellow and the black brown cells are absent, and the color is solely due to the reflective iridophores. Chen's work was really the first proper scientific attempt to understand the physical basis of color in metallic-scaled goldfish. The transparent scaled fish, like shubunkins, are a bit more complex genetically and haven't been studied quite as intensely at that cellular level.
It's amazing how the presence, absence, or density of these tiny pigment cells creates such a huge visible range of colors and patterns.
Right.
You mentioned the geekin earlier, or peacock tail. That sounds really unique.
It is indeed very distinctive. The geekin's absolute defining feature is its tail fin. The two halves of the tail spread out very strongly, almost horizontally. Vertically. They lie almost parallel to each other in a near vertical position when viewed from the side. But the key is how they splay outwards when viewed from behind, creating a distinct X shape.
Ah, okay, X shape from behind. That's why it's called the peacock tail.
Exactly. It's also called kujaku, or kujaku in Japanese, which means peacock tail. Sometimes a few small scales can even develop on the peduncle, the part of the body right between the tail fins.
Is it easy to breed?
No, apparently not. Matsui noted that only about 40% of the offspring in a spawning might fully develop this unique tail shape properly. The ideal coloring is also very specific. Deep, solid red fins contrasting sharply with a predominantly white or silvery body.
Red fins, white body. Striking.
But because getting that perfect pattern is rare and genetically difficult, some sources mention that artificial coloring or even minor surgical alterations are sometimes used to enhance the fish for shows or sale.
Ah, cheating.
Well, perhaps enhancing. The Jiken definitely originated in Japan and is highly prized for its unique appearance, but it remains difficult to breed true and consistently, which makes good specimens quite expensive.
So, a beautiful but genetically very tricky variety.
Hmm.
Now, thinking about the Shubunkin again, Matsui had a slightly different idea about its origin compared to the Wakan Demikin cross, didn't he?
Yes, he did. While another researcher, Akiyama, is usually credited with creating the Shubunkin around 1900 by crossing the Calico Demikin, the Wakin, and possibly the Hibuna, a type of red crucian carp.
Okay.
Matsui came to the conclusion, based on his own breeding experiments, that using the walk-in wasn't actually necessary to get the Shubunkin type.
How do you do it then?
He was able to reproduce the Shubunkin phenotype himself simply by crossing the wild goldfish ancestor, the Funa, directly with the Calico Demikin.
Una. That's the wild type.
Yes, the dull-colored wild ancestor. He argued that this simpler cross genetically provides all the necessary ingredients. The wild-type Funa contributes the single tail and the normal, non-telescopic eyes. And these traits are dominant, so they suppress the Demikin's double tail and telescope eyes.
Right.
And meanwhile, the Calico Demikin parent provides the gene for transparent scales and the modeled calico coloration pattern. The fin length, Matsui reasoned, would likely end up somewhere intermediate between the two parents.
So a more direct route to create a Shubunkin, genetically speaking, focusing just on the key contributions needed from each parent. Makes sense. What about the Oranda? Its origin seems to be a bit contentious.
The Oranda's origin is indeed somewhat debated. A very common belief, still stated often, is that it simply arose from breeding a Ryukin and a lionhead together.
Seems plausible, combining the fringed-tail's body and fins with the lionhead's hood.
It does seem plausible. However, Matsui disagreed with this simple explanation. He pointed to an 1800 Japanese drawing of a fish labeled Dutch lionhead, Oranda shigashira, found in Nagasaki, suggesting the type existed before such a cross might have been common.
Ah, historical evidence.
Right. He also noted a key biological difference. While lionheads can sometimes show some variation in their dorsal fin development, like remnants or partial fins, orandas basically never do. They always have a full dorsal fin.
Interesting distinction.
Yeah.
So what did Matsui think?
Matsui suggested that fish resembling orandas might have arisen in a couple of ways. Yes, maybe from crossing Ryankins and lionheads, but also potentially by selectively breeding fringe tails, Ryankins, that naturally developed some extra skin growth on their heads as they got older.
So selecting for spontaneous head growth within the fringe tail line.
Possibly. It's also conceivable that the trait for having no dorsal fin evolved independently in different lines of goldfish, once in the lineage leading to the lionhead ranchu, and maybe not happening in the lineage leading to the oranda. Matsui's own experiments crossing ranchu and Ryukin goldfish produced very complex genetic results, making him doubt that this specific cross could reliably produce the stable oranda type as we know it.
So maybe multiple paths leading to a similar-looking fish, highlighting the complexity of selective breeding over centuries.
Precisely. And since Matsui's time, the oranda has become incredibly diverse, with many subvarieties.
You mentioned a huge increase in variety overall after Matsui's work in 1934, especially in lionheads, orandas, and telescope goldfish. Did Matsui comment on the goldfish he saw developing elsewhere, like in America or back in China?
Yes, he did make some interesting observations. He noted the American comet's similarities to a Japanese goldfish called the Tetsugyo, though the exact relationship wasn't clear. He definitely considered the American veiltail, developed from the Ryukin, to be a distinct and significant variety in its own right.
And the Shubunkin.
He observed, perhaps a little wryly, that the Shubunkin, despite being developed in Japan, wasn't actually particularly highly valued there at the time, but it was becoming much more popular in the West, particularly in Britain and America.
Funny how tastes differ.
Isn't it? And subsequent breeding efforts, especially in Britain, have actually led to 3 distinct styles or standards for the Shubunkin: the original Japanese style, and the newer London and Bristol Shubunkins, which differ mainly in fin shape.
We'll have to come back to those later.
Yeah.
What about Chinese varieties Matsui knew?
He listed several key Chinese varieties, all of which, interestingly, were also mentioned or described by Chen back in 1925. This included the pearl scale, the pompon, the bubble eye, the outturned operculum fish we mentioned, and distinct color varieties like blue and brown goldfish, plus a variant of the bubble eye called the toadhead. He also noted Chen's observation about bubble eyes, how the development of those fluid-filled sacs under the eyes could sometimes push the eyeball itself upwards, making it look somewhat similar to a celestial, even though the underlying eye structures are quite different.
So different regions were clearly developing their own preferences, focusing on different traits, leading to these distinct strains and variations even back then. And even within a single variety, like the lionhead, you said there's a lot of complexity.
Indeed. The whole lionhead ranchi situation is quite intricate. You have different broad categories, like the red cap oranda, which is technically an oranda but sometimes gets lumped in discussion. Then within the true dorsalis hooded types, you have the lionhead group itself and distinct Chinese lionhead types. And the lionhead group, particularly as classified by Japanese breeders, is further divided into several subtypes. The classic Ranchu, the Osaka Ranchu, the Nankin Ranchu, the lionhead, sometimes referring to the Chinese type, and the Edo Nishiki.
Wow, that's a lot of names. What are the key differences?
Well, a major point is that significant head growth, the hood, really only develops properly in the classic Ranchu, the Japanese standard, and the lionhead, often referring to the Chinese standard. The Edo Nishiki, for example, was basically an attempt to create a calico Ranchulian head, but it generally doesn't develop very impressive head growth.
Why not?
Good question. It might be due to the genetics of the calico pattern interfering somehow, or maybe the lines used just didn't carry strong genes for hood development. The Osaka Ranchu was known for a very rounded, globular body, the specific webbed tail shape, and a broad head, but it's apparently very rare now, possibly extinct.
And the Nankin?
The Nankin, also sometimes called the eggfish, is quite different. It has a smooth back, no dorsal fin, but typically doesn't develop any head growth. It often has a narrower, more pointed head shape compared to the Ranchu, and a common coloration is silver or white with patches of red, especially on the fins and mouth area.
So a hoodless, dorsalis fish.
Exactly. And going back to the Edo Nishiki, or Edo calico, the fact that this fish, often resulting from crosses involving lionheads and possibly calico orandas, has relatively poor hood growth might actually support the idea that the genes responsible for hood development in orandas, which do have good hoods, and ranches evolved somewhat independently.
Interesting genetic puzzle. Any other related types?
There's the Hamacalico, or Hamashiki. This apparently arose from a line of pearl-scale goldfish that also showed goose-like head growth. The Hamanishiki features paired, bubble-like grates on its head, but combined with pearl scales. It's quite exotic.
Pearl scales and head bubbles. It really is like they're mixing and matching all these different traits. You mentioned another author, Tyke Fisher. How did his work add to this picture, especially more recently?
Bernhard Tyke Fischer's book, Goldfish in aller Welt, Goldfish in all the world, became a really valuable resource, especially for Western enthusiasts, because it provided more insights into developments happening in China, particularly after communication became easier. His work essentially builds upon and helps organize the vast amount of information, drawing heavily on Matsui's earlier classifications.
So he synthesized things.
Yes, and he strongly emphasized China's current dominance in developing new and fascinating varieties. He noted that Japan, which was a major innovator for centuries, had shifted its focus much more towards breeding koi carp after World War II.
The koi boom.
Right, so Tyche Fisher established three main lineages or groups to categorize the diversity. Single-tail, fringe-tail, which includes twin tails but with a dorsal fin, and dorsalless twin tails, no dorsal fin. When dealing with crosses like shubunkins, he generally categorized them based on the most distinctive parent variety involved in their creation.
Okay, that sounds like a practical system. What varieties fall into his fringe-tail group, for example? That sounds like a big one.
It is. Tyche Fisher's fringe-tail lineage includes the basic fringe-tail, like the Ryukin, the fan-tail, the Watonai, a sort of intermediate between Wakin and Ryukin, the veil-tail, the goosehead oranda, the standard oranda, the Japanese Narial bouquet or Hanafusa, which has a dorsal fin, the iron fringe-tail, a bronze-colored fringe-tail, the meteor, tailless fringe-tail, the Tosikin with the unique flat spreading tail, and the ironfish, bronze common goldfish.
Wow, that's diverse. What about single-tailed fish that came from twin-tailed lines, like maybe rejects from breeding?
He accounted for those too. He listed single-tailed variants that arose from twin-tailed stocks or specific crosses. This includes the American-Japanese shibunkin, the London shibunkin, the Bristol shibunkin, the ironfish, again if it came from twin-tail stock, and the nymph, which is essentially a single-tailed fantail.
It's fascinating how they tried to bring some order and logic to this incredible, almost overwhelming diversity.
Okay, Let's move firmly into the more modern era. How have goldfish varieties and our preferences for them changed, say, since World War II?
Well, the period after World War II saw a huge increase in the general availability of most of the major goldfish types from the Far East and the West. Before that, many were quite rare or unknown outside Asia.
Right, imports became easier.
Exactly. Alongside that, we saw the development of many subvarieties, especially within the Oranda and telescope groups. This led to what you could almost call variety families. Think of all the different color patterns within the telescope group. Butterflies with tail fins spread like wings. Pandas, black and white. Magpies, mottled black, white, gray.
Lots of descriptive names. Yes. And similarly, many new forms of lionheads and orandas appeared. A more recent trend, as we touched on, is deliberately combining features from different basic varieties. You might see celestials bred with pompons, or breeders adding outturned gill covers, that trait Chen described, to various types like orandas or telescopes.
Looking the boundaries.
Definitely. Although some purists might question whether adding too many extra features actually makes the fish less aesthetically pleasing, maybe a bit overloaded. Interestingly, the Goldfish Society of Great Britain, GSGB, actually has rules against showing fish that have extra features not specified in the standard for that particular variety.
Ah, so the official bodies try to maintain some purity for each type.
They do, yes. It helps keep the distinctions clear.
It sounds like breeders were really getting creative, sometimes creating entirely new combinations of traits. Let's talk about the standards for some of the more common varieties.
Yeah.
What makes a perfect common goldfish, according to the show bench? It seems simple, but is it?
It seems simple, but achieving perfection is hard. The standard for a common goldfish calls for a long, slender body, flattened slightly from side to side. All the fins should be single, one dorsal fin on top, one anal fin underneath, paired pectoral fins, the arms, and paired pelvic fins underneath further back, and a single tail fin.
And the tail shape.
The tail should have a spread roughly equal to the body's depth at that point, with somewhat rounded lobes. The length of those lobes should be about a quarter of the fish's total body length. Critically, the body's curves along the back and belly should be smooth and fairly equal, and the sides should also have a smooth, even contour when viewed from above. No lumps or bumps.
What about color? Anything goes.
The scales should be metallic, providing that shine. The color itself can be red, orange, yellow, blue, brown, black, or silver, either as a single solid color, self-colored, or in a variegated pattern, patches of color. The fins should ideally match the body color or provide a nice contrast.
So quite specific even for the common type.
Absolutely, and it's worth noting that a truly perfect common goldfish, meeting all those points, is actually quite rare and highly valued by breeders and judges. A top-quality common goldfish can absolutely win major awards at shows, competing against the fancier varieties.
Good to know. What about the comet standard? You said it needs to be graceful?
Yes, the ideal comet should embody grace and be very active. It needs that streamlined, slender body, more so than the common goldfish. And the key feature is that deeply forked tail fin, the caudal fin, with pointed lobes that are ideally as long as or even longer than the body itself.
Longer than the body.
Yes, and it should be held nicely spread in a V shape without drooping. Unfortunately, as I mentioned, many comets sold commercially, especially the cheaper ones, often don't meet this standard. Their bodies might be too deep, too chunky, and their tail fins disappointingly short.
So look for the long fins and sleek body. What colors?
The original comets were usually a solid red or orange, but Sarasa comets, which have a red and white patterned body, have become very common and popular. There's definitely potential for dedicated breeders to produce really high-quality comets, perhaps even exploring versions with different scale types, like mirror-scaled comets, or maybe even developing that rumored twin-tailed Yamagata comet version.
It sounds like there's still room for improvement and innovation, even with these more established, seemingly simple varieties. Now, the Shubunkin you mentioned it has evolved into multiple standards, particularly in the UK.
Yes, the Shubunkin isn't just one single type when it comes to how it's judged at shows, especially in Britain. They have separate, distinct standards for the London Shubunkin and the Bristol Shubunkin. Interestingly, they don't have a separate standard for the original Japanese form anymore. North America has also developed its own distinct Shubunkin style, generally based on the Japanese original, but perhaps emphasizing blue coloration more.
So what unites all Shubunkins?
What all Shubunkins have in common is their scales and color pattern. They don't have the fully reflective metallic scales like a common goldfish or comet. Instead, they have mostly transparent scales, which gives them that nacreous or mother of pearl sheen.
Ah, the calico look.
Exactly. And ideally, they should have a multicolored, strongly mottled or variegated color pattern, typically a base color like blue, overlaid with patches or speckles of red, orange, yellow, brown, black and white, or silver. Their body shape is generally similar to the common goldfish, elongated and streamlined.
So the main differences between the styles London Bristol are in their fins.
Precisely. Fin shape and length are the key differentiators. The original Japanese shibunkin has fins that are longer than a common goldfish's, somewhat similar in shape to the comet's, but with a tail fin that's only a little over half the body length, and moderately forked.
Right.
The London shibunkin has shorter, more rounded fins overall, particularly the tail fin, which is less deeply forked and has very rounded lobes. It's closer to the common goldfish finnage.
And the Bristol, that sounds distinct.
The Bristol shibunkin is perhaps the most dramatic. It's known for its very large, well-developed tail fin with broad, rounded lobes, often described as resembling a heart shape when viewed from behind. It should be carried high without drooping. It's a very popular showfish in the UK.
So fin shape really is the key difference between those shibunkin styles.
Yeah,
And you mentioned the calico coloration has some interesting genetics behind it. Not breeding true.
that's right. The desired calico pattern and nacreous sheen in shibunkins are typically the result of the fish being heterozygous for the transparent scale gene. That means it has one copy of the gene for transparent scales, let's call it T, and one copy of the gene for normal metallic scales, let's call it T. So its genotype is TT.
Heterozygous, okay, one of each.
Exactly. And because of this, calico TT shibunkins don't braid true. When two calicos, TT reproduce, Mendel's laws tell us you'll get offspring in roughly these proportions. About 25% will be homozygous metallic, TSG, they'll look like normal goldfish, not calico. About 50% will be heterozygous, TT, they'll be calico like the parents. And the remaining 25% will be homozygous transparent, TT.
TT, what do they look like?
This TT group is often referred to as matte. They lack most of the reflective guanine, and they generally have a reduced amount of all three types of pigment cells: melanin, xanthophylls, guanine. They often look pinkish or pale, sometimes almost colorless, especially when young.
So traditionally seen as undesirable rejects.
Historically, yes, often. However, this reduction in pigment isn't always complete, and by carefully selecting and breeding these matte TT individuals over generations, breeders have actually managed to produce some highly colored true breeding forms that are homozygous transparent. Examples include the sky blue shibunkin, a matte fish with enhanced blue pigment, and the midnight shibunkin, a matte fish that's predominantly black.
Ah, so they can be colored.
Yes, if the underlying pigment potential is there and selected for. This shows real progress in creating shibunkins that can potentially breed true for certain desirable characteristics, moving beyond the limitations of the heterozygous calico state.
That's a significant step forward in breeding for that specific look.
Yeah.
Okay, what about the walkin? You mentioned it as a parent of the shibunkin and the ancestor of fancy goldfish. What's its standard?
Right. The wakin is fundamentally a long-bodied, twin-tailed fish. It really is considered the ancestor, or at least the earliest form, from which all the other twin-tailed fancy goldfish varieties we see today were developed.
So historically very important.
Hugely important. In Japan, it's still highly prized, particularly for the intensity of its red coloration. Wakins with deep red patches on a white background, Sarasa pattern, are especially valued.
Are they always metallic?
Typically, walk-ins have metallic scales, yes. They can come in various colors: red, red and white, orange, white. A calico version with the walk-in's long body and twin tail was produced and marketed as the Edo-Jakin after World War II. It was quite attractive, but it never became hugely popular, partly because some purists felt its tail shape wasn't truly Jikin-like, despite the name. Still, the walk-in itself deserves a place of honor in goldfish history.
Definitely sounds like a foundational variety, valued for its intense red hues back in Japan. Now, the Jikin itself, with that unique X-shaped tail, it must have a very specific standard focusing on that tail and its color.
Absolutely. The Jikin standard is all about that unique tail and the color contrast. The key feature is that splayed twin tail, held almost at right angles to the body when viewed from behind, looking somewhat dumbbell-shaped from the side, creating that X.
And the color pattern.
The ideal pattern is very specific. Deep, solid red on all fins, tail, dorsal, pectorals, pelvics, anal, and sometimes the lips or gill covers contrasted against a body that is predominantly pure white or silvery. Any red patches on the main body are considered faults.
Wow, that's demanding.
Extremely. And it's notoriously difficult to produce perfect geekins that meet this standard. Both the precise tail shape and that specific color pattern aren't easily or reliably inherited. That's why, as we mentioned, attempts to artificially enhance their appearance, like coloring fins or removing body spots, have sometimes occurred, though they're generally frowned upon by serious breeders.
So maintaining the purity of that unique tail shape and color pattern is a real challenge.
It is. This variety really remains one for dedicated specialists who are constantly working to improve the consistency of its key features and perhaps refine the smoothness of its body shape, which should still be relatively elongated like the walk-in.
It sounds like a constant uphill battle for Geekan breeders. Okay, what about the fantail? That sounds more straightforward and it's very popular, right?
Yes, the fantail is much more common and generally easier to breed and keep. You can think of it as a development from the wacken, but with a significantly shorter, more rounded or egg-shaped body and noticeably longer fins than the wacken.
Shorter body, longer fins.
Right. The curves along its back and belly should be smooth and almost symmetrical. It's a deservedly popular and quite hardy fish, suitable for both aquariums and, often, ponds. It comes in both metallic and calico forms.
Is one hardier?
Generally, the metallic fantails are considered a bit hardier than the calicos. While all the standard metallic colors can occur, red and white, Sarasa, are probably the most common, followed by the calico patterns.
So a good, sturdy, all-around fancy goldfish, it seems. Now, the Ryukin, which you said gave rise to the veil tail, its key feature, is that characteristic hump, right? What else defines its standard?
Exactly. The Ryukin standard calls for a short, very rounded, almost globular body, distinguished by that prominent dorsal hump that rises quite sharply just behind the head.
Very noticeable.
Oh, yes. It also has a double tail fin, a caudal fin, that is strongly split and typically droops downwards. The tail fin should be quite long, ranging anywhere from 3/4 of the body length up to 1.5 times the body length.
Quite variable, then.
It can be, yes. The lobes are often rounded, and the whole tail can spread wider than the body itself when viewed from behind. The dorsal fin is single and should be held high, reflecting the hump.
And colors.
Common colors include solid red, orange, red and white, sarasa, pure white, and also calico. It's worth noting that this modern description emphasizing the strong hump is somewhat different from some earlier Western descriptions or illustrations that showed Rikins with a much smoother back contour, more like a fantail with longer fins. The hump seems to have become much more pronounced in the breed standard over time.
That hump is definitely its most recognizable trait today.
Okay.
And the Tosakan, another Japanese variety with a very distinct tail you mentioned.
Yes, the Tosakan is quite special. It also has a short, rounded body, similar in depth to a Ryukin, but its absolutely defining feature is the unique tail fin. It's a fused twin tail that spreads out completely horizontally, like a flat fan, and the front edges actually curve forward towards the head.
Wow, completely flat and spreading.
Yes. It doesn't hang down like a Ryukin or veil tail. It has a very characteristic flowing, undulating shape when the fish swims. The tail's length is usually around 2/3 of the body's length.
What colors does it come in?
Common colors are typically metallic red, orange, and red and white. The curve along the back might show a slight hump, similar to a Ryukin, but really the main focus for breeders and judges is achieving that perfect, unique, horizontally spreading tail conformation. It's considered quite difficult to breed and maintain.
That flowing, flat tail almost sounds like a piece of delicate fabric trailing behind it. Beautiful. What about the veil tail standard? Developed from the Ryukin, you said, but it's all about those long, flowing fins, isn't it?
Absolutely. The veil tail standard is the epitome of long, flowing finnage in goldfish. It emphasizes a deep, rounded body, similar to the fantail or Ryukin, but ideally with that smooth dorsal contour, no hump, and then very long, flowing fins, all of them, but especially the tail fin.
How long?
The tail fin should be at least 1.5 times the body length, and ideally double, flowing down like a veil. The broad tail form, with minimal split between the lobes, is particularly prized over the more deeply split ribbon tail form.
Sounds magnificent. Easy to achieve.
No, maintaining top quality, especially in calico veil tails, has apparently been a significant challenge for breeders almost ever since the standard was first established. While excellent red metallic veil tails are still seen relatively frequently at shows, perfecting the calico veil tail with both ideal color pattern and superb finnage seems to be much harder.
It sounds like achieving and maintaining that extreme level of fin development without sacrificing health or body shape is a constant effort for veil tail breeders. Now, after covering all these more established varieties, you mentioned a huge burst of new Chinese varieties appearing more recently.
Yes, it's really quite something. What has happened in China, particularly in the last few decades, is truly remarkable. The sheer number and novelty of new varieties that have been developed and exported is breathtaking. They've really put themselves back at the forefront of goldfish innovation.
What do you think is driving this creative explosion? Is it just different aesthetic preferences or new breeding techniques?
It's likely a combination of factors, possibly different aesthetic sensibilities, a willingness to experiment and combine different traits in novel ways, maybe large-scale breeding operations allowing for more selection, and perhaps even rediscovering or refining techniques.
Thinking about specific traits, the development of telescope eyes, for example. Is that straightforward? Do they just pop out?
It's an interesting process. When the fish are very young, their eyes look completely normal. But then, usually during the first year of life, they gradually start to protrude outwards.
Gradually.
Yes, it's not instantaneous. And crucially, this development isn't always even between the two eyes. One might develop faster or larger than the other. Breeders have to be quite rigorous in culling fish, removing them from the breeding program if they show uneven eye development. The ideal is large, equally sized, and shaped protruding eyes.
So it's a gradual change that needs careful monitoring by the breeder. What about the bubble eye? Those sacs under the eyes look so fragile.
The bubble eye standard requires a body shape similar to the lion head, short, rounded, and crucially lacking a dorsal fin. The defining feature, of course, is those large fluid-filled sacs or vesicles, the bubbles that develop under each eye. Because of the bubbles, the pupils themselves are usually oriented upwards, similar to a celestial, though the mechanism is different.
What colors are common?
They come in the usual range: metallic, red, orange, white, red and white, and also calico. There's also a less common variant called the toadhead, which has similar bubbles, but they tend to be smaller and perhaps firmer. And yes, you're right, the bubbles are extremely delicate and can be easily punctured or damaged, which can lead to infection or deflation. They require very careful handling and specific tank conditions.
They definitely look like they need extra gentle care. And the pompon, with those fleshy growths on their nose area, how does that work?
There are actually two main forms traditionally recognized. The one most common in the West is dorsalis, like the bubble eye or lion head. But there's also a Japanese form called the hanafusa, which does have a dorsal fin like an oranda, but with pompons.
Okay, but the key is the nose growth.
Yes. The defining feature for both is the hypertrophied nasal septa, basically. The flaps of skin around the nostrils become greatly enlarged, forming these ball-like or tassel-like appendages. The pompons or narial bouquets on the top of the head between the eyes. Ideally, these should be large, well-matched in size, and look quite firm.
And you mentioned the Chinese, like adding these to other fish.
Yes, there seems to be a tendency in modern Chinese breeding to add pompons as an extra feature to many other existing varieties. You might see telescope eyes with pompons, celestials with pompons, orandas with pompons. But as with adding too many features, combining prominent pompons with other head growths like hoods or bubble eyes can sometimes make the fish look a bit cluttered or overdone, aesthetically speaking.
So sometimes keeping it simpler might be better when it comes to fancy head features.
Well, yeah.
Okay, the Oranda standard has the hood, of course. What else defines it?
The Oranda standard generally describes a short, deep, rounded body, quite similar to a fantail or ryukin, but it must have a dorsal fin. It has a double tail fin, which can be moderately long and flowing, either forked, like a fantail, or ideally broad tail, like a veil tail. And the most prominent feature is, of course, the fleshy hood, or wen, that develops on its head.
Where exactly does the hood grow?
The hood growth can occur in three main areas. On the top of the head, cranial growth; on the gill covers, opercular growth; and in the area below and around the eyes, infraorbital growth. High-quality modern orandas often show good development in all three areas, creating a full hood.
Are there different types of Oranda based on the hood?
Yes, there are often different standards or classes for orandas with full hoods covering most of the head versus those that only have growth on the top of the head, like a cap. Those are often called red cap orandas if the cap is red and the body is white, or tanto orandas more generally.
And they get big.
Orandas have the potential to grow quite large, becoming very impressive specimens. And as we've alluded to, breeders, particularly in China, have recently developed many novel color combinations, especially involving the hood. You might see black hoods on red bodies, white hoods on red bodies, red hoods on black bodies, even multicolored or patterned hoods, though the stability of some of these very novel color combinations over the long term remains to be seen.
They really do come in a stunning range of hood colors and patterns now. Okay, and finally, the Ranchu lionhead standard. How is that distinct from the Oranda, besides lacking the dorsal fin?
Right. The Rancho Lionhead standard describes a fish with a short, deep, rounded body, but importantly, no dorsal fin. There's a key difference in the back profile often emphasized between the Japanese Ranchu and the Chinese lionhead. The Ranchu standard calls for a strongly curved back contour, while the Chinese lionhead often has a straighter, flatter back profile.
Curves back versus straighter back.
Yes. Both have a double, typically forked tail fin, which is usually held more horizontally or slightly upwards compared to the drooping tail of a Ryukin or veil tail, and it generally doesn't sweep downwards. And of course, the other key feature is the hood development, which is the main variable within this group.
How does the hood differ from the Orandas?
The development of the hood in the Ranchulian headline seems to have followed a somewhat different path historically than in the Oranda. For instance, the goose-like head growth, sometimes seen in older Oranda types, is less common in Ranchu. A significant difference has also evolved, particularly emphasized by Chinese breeders, between types they call tiger heads, which have a smooth, relatively uniform hood development, and lion heads, where the hood growth in those three distinct regions, cranial, opercular, infraorbital, is more pronounced and lumpy, sometimes even differing in color or texture between the regions.
So even the type of hood growth is specified. It's clear that what we consider beautiful in goldfish and the standards that attempt to define these ideals are incredibly complex and seem to be constantly evolving. These standards aren't just for judging fish at competitions, are they? They must be important for breeders, too.
Oh, absolutely essential. Show standards serve as crucial benchmarks or guidelines for breeders. They help them decide which fish to select for breeding, aiming to produce offspring that more closely match the ideal for that variety. It guides their selection objectives.
And presumably, better fish fetch better prices.
For commercial breeders, yes, producing fish that meet the show standard, especially high-quality examples, can certainly command a higher price. But even for hobbyist breeders, the standard provides a target to aim for, a way to measure progress in improving the overall quality and consistency of their fish line. Having clearly defined, widely agreed upon standards is really important. The veil tail is a good example. Its core features have remained fairly consistent over a long time, providing a stable goal.
Were the original standards simpler?
The initial goal of the Goldfish Society of Great Britain, GSGB, when they first started setting formal standards was actually to define a limited number of distinct basic varieties. They wanted varieties that wouldn't easily interbreed to look like something else, and that didn't rely on too many overlapping special characteristics. This was partly to prevent lower quality fish from being passed off or mislabeled as different, more valuable varieties.
Makes sense. Has that become harder?
Yes, as the number of recognized standard varieties has grown significantly, applying those original principles has become more challenging. We now see the emergence of what some call identikit standards, where, for example, the standard for a broadtail Moor or a broadtail Oranda is essentially the standard for a veil tail, just with telescope eyes or a hood added.
So like a basic body plan with optional extras.
Pretty much. And similarly, the body standards for bubble eyes, celestials, and pompons often closely resemble the lionhead ranchu standard, dorsalis short body, just with their specific eye or nose feature added. This proliferation of similar standards with minor additions can create difficulties for judges trying to differentiate nuances, and it might not always serve the distinct goals of breeders focusing on perfecting those core body shapes versus the added features.
The standards might need revising.
Definitely. The evolution of fantail styles over time, for instance, also highlights the need to periodically review and update standards to reflect how the fish themselves are changing through breeding.
How should we appreciate these fish then? Just side view.
Ideally, appreciating goldfish fully involves looking at them from all angles. The side view is traditional in the West for assessing body shape and finnage. But viewing from above is also crucial, especially in the East, China and Japan, to appreciate the body width, symmetry, and how the fins are carried. And beyond the physical points of the standard, overall condition and deportment, how healthy, active, and gracefully the fish carries itself, is also a major factor in judging. And of course, the exhibitor plays a role in preparing their fish to look its best for a show.
So a good standard is like a living document that needs to reflect the fish, guide the breeders, and help the judges, but also evolves over time. Okay, let's switch gears again, maybe get a bit more technical. Let's delve into the actual genetics behind improving goldfish. How do breeders consciously use the principles of inheritance to create or refine these amazing varieties?
Right. At its core, improving goldfish through breeding is fundamentally about genetic manipulation, albeit through traditional selective breeding rather than lab work, usually. Understanding Mendelian inheritance, those basic rules discovered by Gregor Mendel about how traits are passed down, is absolutely key.
Mendel and his peas again.
Exactly. His work showed that heritable characteristics are passed from parents to offspring in a predictable, particulate way. He realized that traits often come in contrasting pairs, like yellow versus green peas, and behave as distinct units. These hereditary factors, which we now call genes, retain their identity across generations. They don't blend, they remain discrete. And crucially, they usually exist in pairs within an individual.
The two alleles for each gene.
Precisely. His first big principle, segregation, states that when an organism makes its reproductive cells, sperm or eggs, the two copies, alleles, of each gene separate, so each reproductive cell only gets one copy. His second principle, independent assortment, states that the genes for different traits, like seed color and seed shape, sort themselves into reproductive cells independently of one another, provided they're on different chromosomes.
So the same basic rules that apply to pea plants also apply to goldfish, even with their funky chromosome situation.
The fundamental principles hold true, yes. Segregation and independent assortment still happen. However, as we discussed, having those extra sets of chromosomes in goldfish, being polyploid, adds layers of complexity to the observed patterns of inheritance, the ratios you see in offspring. But the underlying mechanisms are Mendelian.
Can you remind us of Mendel's basic experiment?
Sure, his classic experiments with single traits, monohybrid inheritance, like crossing pure-breeding yellow seed peas with pure-breeding green seed peas, demonstrated dominance. All the first-generation F1 offspring had yellow seeds, showing yellow was dominant over green.
Right.
Then when he crossed those F1 plants together, the second generation, F2, showed both yellow and green seeds again, but in that famous, predictable 3-to-1 ratio, 3 yellow for every 1 green.
3.1 ratio.
Yes, and further experiments, like back-crossing the F1 hybrids to the recessive green parent, revealed the underlying genetic ratio of 1 homozygous dominant YY, 2 heterozygous YY, 1 homozygous recessive Y, which explains why the F2 phenotype ratio is 3.1.
Okay, that covers one trait. What about two?
When Mendel looked at 2 traits simultaneously, dihybrid inheritance, like seed color, yellow-green, and seed shape, round-wrinkled, he observed independent assortment. If you cross parents pure for both traits, e.g. yellow round X, green wrinkled, the F2 generation shows all 4 possible combinations, but again in a predictable ratio. 9 yellow round, 3 yellow wrinkled, 3 green round, 1 green wrinkled. That classic 9.3.3.1 phenotypic ratio is the hallmark of 2 independently assorting genes.
Those ratios are so fundamental to classical genetics. Are there any genetic quirks in goldfish that mess with these ratios, like genes that might be harmful or even lethal, that breeders need to watch out for?
Yes, lethal genes can definitely play a role and alter expected ratios. Sometimes a gene that has a desirable effect when heterozygous, 1 copy, can be lethal when homozygous, 2 copies. A known example in common carp, which might apply to goldfish derived from crosses, involves the gene for mirror scales, very large, sparse scales.
Mirror scales.
In carp, the allele for mirror scales, let's call it N, is dominant over the allele for normal scales N. However, the homozygous mirror genotype, NN, is lethal. Those embryos don't survive. So if you cross 2 mirror-scaled carp, which must be heterozygous, NMXNN, you don't get the expected 1N and 1NN ratio. You only get the survivors, which appear in a 2NN mirror core-NN normal ratio. That 2.1 ratio is often a clue that a recessive lethal gene is involved.
So if goldfish breeders working with mirror scales see that 2.1 ratio, they might suspect the same lethal gene is at play.
Exactly. While goldfish with mirror scales have been produced, likely through crosses with mirror carp, it hasn't been definitively proven if the exact same lethal gene mechanism operates in goldfish, but it's a strong possibility breeders should be aware of.
What about traits that aren't simple on-off, but more like a range, like fin length?
Right, that falls under quantitative inheritance, also called polygenic or multifactorial inheritance. Many traits, probably including things like intensity of color, fin length, body depth, and maybe even hood development, aren't controlled by just one single gene pair. Instead, they're influenced by multiple genes, often on different chromosomes, each contributing a small additive effect to the final phenotype.
So lots of genes adding up.
Yes. This results in a continuous range or distribution of appearances in the population, often following a bell-shaped curve rather than the distinct, clear-cut categories you see with simple Mendelian traits. Breeding for these traits is more about selecting fish that are at the desired end of the spectrum and hoping their combination of polygenes passes on.
Makes sense. Any other genetic interactions?
Sure. Epistasis is another important concept. This is where one gene locus can mask or modify the expression of a gene at a different locus. The gene doing the masking is called epistatic, and the one being masked is hypostatic.
Can you give an example?
Think about coat color in Labrador retrievers. One gene determines black versus brown pigment, but a separate gene determines whether that pigment actually gets deposited in the hair. If the dog is homozygous recessive for the deposition gene, it will be yellow regardless of whether it carries genes for black or brown pigment. The deposition gene is epistatic to the pigment color gene. Similar interactions certainly occur in goldfish color genetics, leading to modified ratios like 9.7 or 9.3.4 instead of the basic 9.3.3.1.
Okay, one gene overriding another. What about genes that are physically close together?
That's linkage. Genes located close together on the same chromosome tend to be inherited together as a block, more often than not, because the chance of a crossover event happening exactly between them during meiosis is relatively low.
So good traits might be stuck with bad ones.
That can happen, yes. A desirable trait might be linked to an undesirable one, like a disease susceptibility or a poor body shape feature. This can make a breeder's job harder. However, linkage isn't usually absolute. Crossovers or recombination events do happen. So, through careful, often large-scale breeding and selection over generations, progeny testing, breeders can sometimes manage to break undesirable linkages and combine the good traits they want.
Requires patience and numbers. What about sex linkage? Traits carried on X or Y chromosomes.
Sex linkage occurs when genes are located on the sex chromosomes, like X and Y in mammals, or Z and W in birds and some fish. This leads to different inheritance patterns in males versus females, and reciprocal crosses, e.g. red male X, white female versus white male X, red female, might give different results. However, so far, no major economically or aesthetically important traits in goldfish have been clearly shown to be sex-linked in this way. Although interestingly, scientists like Yamamoto in Japan did manage to experimentally create YY male goldfish, proving a Y chromosome exists, but standard traits don't seem tied to it.
Okay, so probably not a major factor for most breeders. But the polyploidy, those extra chromosome sets, is a big factor, right?
Absolutely. Polyploidy is fundamental to goldfish genetics. As we established, they are tetraploid 4N, likely having evolved from a diploid 2N ancestor that underwent genome duplication. So instead of 2 copies of each chromosome, they have 4. Or more accurately, they have 100 chromosomes, likely representing 4 sets of an ancestral genome that had 25 chromosomes. 4 by 25 equals 100.
And this changes the inheritance patterns.
It does. The exact genetic consequences depend on how those 4 sets of chromosomes pair up during meiosis. If it's considered allopolyploidy, where the duplicated sets came from slightly different ancestral species and behave more like 2 distinct diploid sets, you might see duplicate inheritance for some genes, potentially leading back to simpler ratios, or maybe 15.1 ratios for single dominant genes. If it's closer to autopolyploidy, where the sets are highly similar from the same species, the pairing can be more complex, multivalent pairing, leading to even more complex ratios, sometimes approaching 35.1 for a single dominant gene. Understanding which model best fits goldfish is crucial for predicting inheritance accurately.
So knowing whether those extra chromosomes are treated as 2 pairs or one big group of 4 makes a big difference for predicting offspring. Wow. What about genetics at the population level? How does that apply to someone breeding goldfish in their tanks?
Population genetics principles are very relevant. The Hardy-Weinberg equilibrium is a theoretical baseline. It states that in a large, randomly mating population with no mutation, migration, or selection, the frequencies of alleles and genotypes will remain constant from generation to generation. The formula POP plus 2PQ plus Q plus 1 describes this, where P is the frequency of one allele and Q is the frequency of the other.
But goldfish breeding isn't random mating in a huge population.
Exactly. Goldfish populations under human care are usually not large. Mating is definitely not random. Breeders choose pairs. Mutation happens, though slowly. And there's intense selection for desired traits. All these factors cause deviations from Hardy-Weinberg equilibrium. They are the very forces breeders use to change gene frequencies over time.
So breeders are actively disrupting the equilibrium.
Precisely. For example, even if a recessive allele for a desirable trait, like maybe telescope eyes, is initially rare, low Q value, practices like inbreeding, mating-related fish, increase the chance of producing homozygous recessive individuals, Q, making the trait appear more often. Selection then favors those individuals, further increasing the frequency of the recessive allele cue in the breeder's line over generations.
So they're intentionally manipulating the genetic makeup of their fish populations over time. What are some specific breeding techniques they use to achieve this?
Back crossing is a very common and powerful strategy, especially when you have one particularly outstanding individual fish.
The starfish.
Right, the elite parent. The breeder might cross this starfish with another fish from a similar good quality line. Then they select the best offspring from that first cross, the F1 generation, and breed them back to the original star parent.
Breeding children back to the parent.
Yes, this is the first back cross producing BC1 F1 offspring. The goal is to concentrate the desirable genes from the star parent into the subsequent generations while still maintaining some genetic diversity from the other line. You might repeat this process for several generations, always selecting the best offspring to cross back to the elite ancestor or its best descendants.
Trying to lock in those good traits.
Exactly. But alongside this, progeny testing is absolutely crucial. You need to see how well a breeder's potential breeding fish actually pass on their desirable traits to their offspring. Even a champion fish doesn't always produce champion offspring consistently, so you have to assess the offspring to know if the parent is genetically valuable.
Don't put all your eggs in one basket, genetically speaking.
Right. And selecting breeding pairs that complement each other is also smart. Maybe one fish has excellent body shape but just okay color, and another has fantastic color but slightly poorer shape. Breeding them together might produce offspring that are good in both aspects. This also helps maintain genetic variation within the line.
But isn't there a risk with all this close breeding, like back-crossing, inbreeding problems?
Absolutely. Inbreeding is a double-edged sword. Some level of it is necessary to fix desired traits and make a line breed true. But excessive inbreeding increases the chances of offspring inheriting two copies of harmful or deleterious recessive alleles that might have been hidden in the heterozygous state.
So you get deformities or sterility.
Yes, that's the risk. You can end up linking the desirable alleles you want with these detrimental recessive ones, leading to lines that become unhealthy, infertile, or deformed over time. It's called inbreeding depression.
How do breeders manage that?
Good record keeping is essential. Tracking pedigrees to avoid mating very closely related fish too often. One strategy to circumvent severe inbreeding issues while still making progress is recurrent selection. You might try to fix desirable alleles in several different, less related lines concurrently. Then you can cross these improved lines together periodically to bring all the good traits together through recombination, while hopefully reducing the frequency of any specific undesirable genes that might have built up in any single line.
Sounds complex. What about newer technologies?
Biotechnology offers some interesting possibilities, though it's perhaps less common in hobbyist circles. Gynogenesis, which we discussed earlier, inducing development of eggs using irradiated sperm, so the offspring are clones of the mother, has generated commercial interest in aquaculture as a way to efficiently produce large numbers of genetically identical, high-quality fish, for example, all-female populations.
Where breeders use that?
Potentially, yes, to rapidly multiply the genetics of an outstanding female. However, you have to be aware of potential pleiotropic effects, where a gene selected for one trait might have unintended side effects on other traits. Biotechnology can also be a positive tool for research, like using comparative biochemical studies, analyzing proteins or DNA, to better understand how different goldfish varieties evolved and how they relate to each other.
So technology offers tools, but the basic principles of selection and managing genetics remain key.
Absolutely. Ultimately, successful long-term goldfish improvement relies on understanding population genetics, maintaining a healthy gene pool, being aware of things like the founder effect when a new line is started from very few individuals, and genetic drift, random fluctuations in gene frequencies, especially in small populations, and carefully managing inbreeding. It's a continuous balancing act.
It really is a fascinating blend of art and science. Now let's connect some of this genetics back to specific, really important mutations in goldfish history. The xanthic mutation, for instance, the one that gives us the reds, oranges, and yellows by removing the dark pigment. You said it's unusual in goldfish.
Yes, the xanthic mutation in goldfish has some quite unusual features compared to similar color mutations in many other fish species. Firstly, and most significantly, it behaves as a dominant gene in goldfish.
Dominant. So you only need one copy to lose the dark color.
Correct. Whereas in many other fish, like zebrafish or medaka, the common xanthic or golden mutations are typically recessive. You need two copies to see the effect. Secondly, the timing of its expression in goldfish is interesting. It usually appears in juvenile fish, often around 2 months old when they lose their initial dark, wild-type coloration, a process called depigmentation or decoloration. But sometimes this change can be significantly delayed, happening much later in the fish's life.
Why would it become dominant in goldfish?
It's been suggested that the dominance of this trait is actually a result of over 1,000 years of artificial selection by humans. People actively preferred and selected the brighter, non-camouflaged colored fish. This intense selection pressure could have favored genetic modifiers that made the xanthic phenotype express itself even when only one copy of the mutant gene was present, effectively making it dominant over time.
Wow, so human preference literally flipped the genetic behavior of this color trait in goldfish compared to its wild relatives or other fish. Amazing.
It's a strong possibility. And even in those feral goldfish populations we talked about, the ones that have escaped and established themselves in the wild, this xanthic gene might persist if the fish only lose their dark pigment relatively late in life, after they've already survived the vulnerable juvenile stages and possibly reproduced. Or perhaps in turbid, murky water, the lack of camouflage is less of a disadvantage.
Makes sense. Okay, what about the twin tail mutation? That's so fundamental to almost all the really fancy varieties. You said it's unique.
The twin tail mutation is considered really quite unique in the world of domesticated ornamental fish. It's been known for roughly 500 years, based on historical records, and nothing truly comparable seems to have emerged and become established in other commonly kept ornamental species like guppies or bettas.
How does it work genetically? Simple dominant.
Genetic studies suggest it's controlled by a single major gene that acts as a dominant but with incomplete penetrance.
Incomplete penetrance, meaning
Meaning that even if a fish inherits the gene, it doesn't always show the twin tail trait. Whether it does or not seems to be strongly influenced by environmental factors, particularly the water temperature during a critical period of embryonic development.
temperature affects tail development.
Yes, higher water temperatures during that sensitive window significantly increase the penetrance, the likelihood that the twin tail trait will actually appear in fish carrying the gene. Lower temperatures decrease the chance.
That's fascinating. What does the gene actually do?
It's thought that the twin tail gene causes some kind of developmental instability, primarily affecting the formation of the median fins, the caudal, tail fin, and the anal fin underneath. It leads to that duplication or splitting of the tail and often the anal fin too. And the same developmental instability might also contribute to the frequent loss or reduction of the dorsal fin in many twin-tailed lineages like lionheads and bubble eyes.
So one gene has a ripple effect on multiple fins.
It seems so, primarily the unpaired median fins. Interestingly, the paired fins, the pectoral fins at the front and the pelvic fins underneath, don't seem to be affected by this particular mutation. They remain single.
So that single gene, influenced by temperature, is responsible for the defining feature of fantails, rayukins, arandas, moors, lionheads. Incredible impact. And you mentioned the evolution of dominance again for both xanthic and twin-tail traits.
Yes, the fact that both the xanthic, color loss, and twin-tail mutations appear to have become effectively dominant in goldfish, unlike their typical behavior in other species or presumed ancestral state, is likely a consequence of long-term artificial selection. Breeders would have consistently selected fish showing the desired traits, bright color, double tail, even if they were heterozygous. Over many generations, this selection likely favored the accumulation of other genes, modifier genes or polygenes, that enhanced the expression of the mutant phenotype when even just one copy of the main mutant allele was present.
So other genes boosted the effect of the main gene.
Exactly, effectively making the main gene behave as dominant within the genetic background of domesticated goldfish. There was an interesting experiment cited where a red metallic veil tail, which should carry dominant twin-tail genes, was crossed with a wild crucian carp, the presumed ancestor with single-tail genes. The resulting offspring were mostly single-tailed.
The wild genes suppressed the twin tail.
It suggests that the genetic background of the wild crucian carp somehow reversed or suppressed the dominance of the twin-tail gene. This really highlights how the overall genetic context, shaped by selection, influences how individual major genes are expressed.
It really underscores how artificial selection by humans has reshaped not just the physical appearance of these fish, but even the fundamental way their key traits are inherited. Amazing. What about how their fins develop after they hatch, especially in those varieties with extremely long fins? That must be important for breeders.
Yes, fin growth, particularly in the long fin varieties like veiltails and comets, generally continues throughout the fish's life, and often the fins grow at a proportionally faster rate than the rest of the body, especially when young.
So long fins early isn't necessarily good.
Counterintuitively, no. If young fish develop extremely long fins very early on, precocious development, it's often seen as undesirable by experienced breeders. They know these fish are likely to become overly finned as they mature, potentially leading to poor swimming ability, fin curling, or reduced fin quality later in life.
So you want slower, more sustained fin growth.
Ideally, yes. For the really top quality long fin varieties, they might not actually reach their peak finnage quality, the best length, shape, and condition until they are 5 years old, or perhaps even older.
So patience is definitely a virtue for breeders aiming for those elegant flowing fins. It takes time to see the true potential. Now let's circle back to color genetics for a moment. You mentioned the three main types of pigment cells derived from the neural crest: melanophores, xanthophores, iridophores. How do specific genetic changes affect these to create the variety we see?
Right. Color variations arise from genetic mutations that affect the number, distribution, or function of these different chromatophore types. A single gene mutation might affect just one cell type or sometimes multiple types. For example, some of the genes Kajishima studied, which cause depigmentation, loss of color, seem to primarily affect the melanophores, reducing black-brown, but might also impact the xanthophores, red yellow to some extent.
What about the iridophores, the shiny ones?
Mutations affecting iridophores are crucial for understanding metallic versus nacreous, gallico-shabunkin versus matte scales. The key gene here seems to be the transparent locus, often symbolized as TT. It acts as an incomplete dominant,
Incomplete dominant.
meaning the heterozygote, TT, looks different from both homozygotes, PT and TT. The homozygous recessive TT condition gives you the normal, fully reflective metallic scales packed with iridophores. The heterozygous condition, TT, causes a significant reduction in iridophores, leading to that pearly, translucent, nacreous sheen characteristic of shabunkins and other calico fish.
And the homozygous dominant TT.
The homozygous dominant TT condition, often called MAT, results in a near-complete suppression of all three chromatophore types, especially the iridophores. This leads to fish with very little or no reflective tissue, often appearing pinkish or whitish due to the underlying tissues showing through.
So that one gene locus controls the whole metallic calico mad spectrum.
It seems to be the major player, yes. The teat calico state, by reducing the surface shine, also allows the colors from the deeper melanophores and xanthophores to be more visible, which is essential for the modeled calico pattern.
Are there other genes affecting shine?
Yes, there appears to be at least one other distinct recessive mutation that also suppresses iridophores. This has been described under different names, like net-like transparent or mock metallic in domestic strains, and it seems to be the same mutation responsible for the bluebelly phenotype found in those feral populations in Ohio.
The blue belly again.
Yeah. In this case, the reduction in iridophores makes deeper-seated melanin pigment more visible, especially on the belly, hence the name. Environmental factors like water chemistry might also influence guanine development, possibly interacting with this gene.
Okay, what about the melanophores, the black-brown cells?
Their absence or reduction, often due to the dominant xanthic gene, which suppresses melanin, leads to the typical colors of a common goldfish: red, orange, yellow. Conversely, an enhancement or increased number activity of melanophores results in blackfish, like the Moor. The interaction between the transparent gene, TT, and melanophores can also contribute to blue coloration in shabunkins.
Can you get true albinos?
True albino goldfish, which completely lack melanin pigment and typically have pink eyes, do exist. They're often called lutinos if they retain yellow-orange pigments. Completely white fish, leucistic, can also occur, often resulting from the interaction of the xanthic gene with other pigment-reducing factors. Albino lutino goldfish have become commercially available more recently, but they haven't gained huge popularity among traditional fanciers yet.
And the telescope eyes. Simple genetics.
The telescope eye mutation, responsible for the protruding eyes of Moors, Demikins, etc., is well established as being caused by a single recessive gene, often symbolized as ID. It's been known since at least the 18th century.
Any other weird scale types?
There's mention of a hammer scale variant, apparently recessive, where the scale surface is concave, curving inwards the opposite of the pearl scale mutation, which has raised convex scales. Hammer scale doesn't seem to have been reported much recently, though. And the mirror scale, as we discussed, likely came from crosses with mirror carp and might involve that recessive lethal N gene, potentially leading to 2.1 ratios if bred amongst themselves.
And the Jigen, just to recap.
Right, the Jiken is essentially a derivative of the Wakin lineage, but selected for that unique splayed tail shape and the very specific red fin silver body coloration. Breeders face constant challenges in maintaining the quality, preventing the red fin color from fading or bleeding onto the body, avoiding excessive body color, and achieving that sharp contrast. That's the goal.
It's truly incredible how many different genetic paths have been explored, accidentally or intentionally, leading to the goldfish we see today. Okay, let's think about putting this genetic knowledge into practice for actual improvement.
Right. Fundamentally, goldfish improvement is genetic manipulation through selection. If a breeder wants to establish a new, superior lineage based on an outstanding fish.
The starfish again.
Exactly. A hypothetical program might look like this. First, cross the elite fish with several individuals from good, related, but perhaps slightly different stock to maintain some variability. Then, select the best F1 offspring.
Yep.
Next, backcross those selected F1 fish to the original elite parent, or its best relatives if it's no longer available. This produces the BC1F1 generation. From these, select the very best individuals.
On what?
You could then interbreed the best BC1F1 individuals to produce an F2 generation from the backcross, BC1F2, select the best from that generation. Then you might repeat the process, backcross the best BC1F2 fish back to the elite line again, making BC2F1, produce a BC2F2, and so on.
Wow, sounds like a lot of generations and careful selection.
It is. It takes time and careful record keeping. This process, repeated over several generations, aims to increase the frequency of the elite parent's desirable genes while gradually establishing a stable breeding population that carries those traits, but hopefully retains enough genetic variability to stay healthy. Progeny testing at each stage is vital to ensure you're actually making progress.
And avoiding too much inbreeding is key.
Absolutely critical. As we said, inbreeding is necessary to fix traits, but it's a double-edged sword. It can concentrate harmful recessive alleles that were hidden, leading to sterile or deformed stocks if not managed carefully. Selecting complementary parents, maybe ones that excel in different aspects of the standard, can help maintain genetic variability and heterozygosity, potentially masking some deleterious recessives.
So balancing fixing traits with keeping the gene pool healthy.
Precisely. Recurrent selection strategies, where you improve different lines somewhat independently and then cross them strategically, can also help circumvent the worst inbreeding problems by allowing recombination to separate linked genes and reduce the frequency of undesirable alleles that might have become fixed in one line.
What about using biotechnology like ganogenesis here?
Ganogenesis, producing maternal clones, could theoretically be used by breeders to rapidly propagate the genotype of an exceptional female fish. It has commercial potential for producing large numbers of uniform, high-quality fish efficiently.
Any downsides?
Well, relying heavily on cloning reduces genetic diversity, which can be risky long-term. Also, we have to be mindful of potential pleiotropic effects. Genes selected for one desirable trait might have unknown negative consequences on other aspects of the fish's health or viability. On the positive side, biotechnology, particularly molecular techniques like DNA analysis, can be incredibly useful for research, helping to clarify evolutionary relationships between varieties and studying genetic diversity.
So, powerful tools, but they need to be used wisely.
Exactly. Understanding population genetics concepts, the importance of maintaining a healthy gene pool, the random effects of genetic drift in small populations, the potential impact of the founder effect when starting new lines, and the risks versus benefits of inbreeding is crucial for anyone serious about the long-term improvement and conservation of goldfish varieties. It all started from a limited sample of wild ancestors, after all.
It truly is a complex interplay of genetics, selection, history, and aesthetics. This has been an absolutely incredible deep dive into the world of goldfish, way beyond the simple orange fish in a bowl. It's truly amazing how much complexity, history, and science lies beneath the surface.
Absolutely. And we've really only scratched the surface today on some of the key aspects, but hopefully we've given you, the listener, a much richer appreciation for their fascinating evolutionary journey, the intricate genetics involved, and the really significant role humans have played and continue to play in shaping the goldfish into the diverse and captivating creatures we know.
Indeed. It definitely leaves you thinking. Considering the centuries-long focus on breeding for specific, sometimes extreme, appearances, what's the right balance between pushing those aesthetic boundaries further and ensuring we maintain enough underlying genetic diversity for the future health and resilience of these fish? That's definitely something to ponder. Thanks for joining us on this deep dive. We really encourage you to explore the captivating world of goldfish further.